Alright guys, today we're going to be talking about DNA replication. But before I get into that, I wanted to do a quick review on DNA that we talked about yesterday. So, as you remember, DNA is made out of a sugar and phosphate backbone, which is represented by that red bar on the bottom where the line's pointing to, and the nitrogenous bases, which we know as our adenine, thymine, guanine, and cytosine. So if we remember, the A and the T in that upper left-hand green box, the A and the T go together and the G and the C go together. We also know that DNA is anti-parallel, which means that it goes from 3' to 5' on one side, and then the other side is just flipped 3' to 5'. We also know that DNA is organized into a double helix, which you see on the right side. Alright, now that we've reviewed a little bit about what we learned about DNA from yesterday, we're going to go on to talk about today's topic, DNA replication. So, DNA replication is literally what it sounds like. Replicating DNA. It's making more copies of DNA so that we can put them into new cells that our bodies have made. So, and as you can see in the picture on the right, our DNA takes the blue strand, which are our original strand of DNA, or our parent strand, as it says in the picture, and it actually splits apart and we make daughter strands, or the red strand, as you can see in the picture. So when we make new DNA, we actually have one old strand and one new strand, and that's called semi-conservative, which is that yellow word that you see there on the screen. Alright, we're going to move on to the next part. So the way that our DNA is able to actually do that splitting apart so that we can make our semi-conservative DNA replication is by this enzyme called helicase, and that's represented by that little purple ball in the middle of our two DNA strands, which are represented by the blue lines. And so that little purple ball, it actually unzips our DNA strand and separates the nitrogenous bases. And that is called our replication fork. Where it's open like that, that's our replication fork. And these words are all in yellow on the side, and the yellow words are what are important, so I would pay attention to those. And so once our DNA is open, and it kind of looks like this bubble in the picture before, you just saw half of this bubble, but when the helicase opens it, it makes this bubble like this. And the DNA isn't able to just be built by itself. It has to have a little help. It has to have an RNA start point. So RNA primase, which is another enzyme, goes in and it makes these little RNA primers, which are literally just little strands of RNA that go on the DNA. So you can see those in green, and they just match up with the DNA opposite of it, of the template strand, and they are these little RNA like start codes. It's kind of like start here. And that's because our DNA polymerase, it can't start on its own. It's just not able to do that. So it needs that help from that RNA primase to do that. So DNA polymerase is a really cool protein or enzyme, which is also a protein, and it works from the 5' to 3'. Always works 5' to 3'. That's really important. That's why that's in yellow. And it's going to build our new DNA strands from that 5' and 3' standpoint. So it's going to start where that little RNA primer is, which actually isn't on this picture, but if you can imagine those greens lines at the beginning of the 5', those little green things that were on the last slide, then you'd be able to remember those RNA primers. And they actually build from those primers, and the DNA builds from that way. And you might notice that it's a little weird. Why are there 3 arrows on one side and one on the other? We're going to get to that. So, once it's done building all of these Okazaki fragments, and now we're going to take that right side, look at that. Those fragments are all separate, and they need to be connected. And that's where the ligase enzyme comes in. And ligase is represented by the yellow ball, And it goes in and it's like, all right, I'm going to connect you now. And it actually connects those strands together so that we have one solid DNA strand. Okay, guys, now let's see if we can remember all of this all together. Okay, I'm going to go through it and we're going to see if we can remember through this overview. So it starts out with helicase. That's the purple ball. And it goes through and it opens up that replication fork. And when it opens up that replication fork, that means that we can start building our new DNA strands. But remember, our polymerase can't start the strands on his own, so he needs help from RNA primase. So RNA primase goes in and he builds the primers. So now once we have our primers, then DNA polymerase is like, yay, I can start. And he goes in and he starts building the new DNA strands, 5' to 3'. On the leading strand, it just goes in one big strand, but on our lagging strand, it's built into Okazaki fragments. And these fragments are broken up into different parts. And so once the DNA polymerase is done, they're still in pieces, which means we need our ligase. So ligase goes in and he connects those DNA fragments into one strand. And that's how we do DNA replication. I'll see y'all tomorrow.